Welcome everybody again to another episode of the ideaXme show, the show that profiles the humans behind the really big ideas that are shaping our world, inspiring the future and future creators, and for all those that like really great stories. I'm Ira Pastor, your life science ambassador, along for the journey today. Today we are going to journey into the always fascinating topic of stem cells and regenerative medicine, and head over to the West Coast of the United States, to San Francisco and the headquarters of the California Institute for Regenerative Medicine, which was an organization that was created originally back in 2004 when voters in California approved a state proposition which allocated $3 billion time to fund this very exciting area of medicine. I'm happy to say that today I'm joined by Dr. Maria Millan, who is President and CEO of the California Institute of Regenerative Medicine. Dr. Millan is a physician and a scientist who has devoted her career to treating and developing innovative solutions for children and adults with debilitating and life-threatening conditions. After receiving an undergraduate degree from Duke, where she started focusing on immunology research, she got her MD here in New Jersey at UMDNJ, and then went on to complete her surgical training and postdoc research at Harvard. After a transplant surgery fellowship at Stanford University, she began her academic career with the pediatric and adult transplant surgery practice. In parallel to that, she continued her bench research at Stanford and became associate professor and director of the pediatric organ transplant program. She served on multiple leadership teams, including the Faculty Senate, the Dean's Faculty Committee at Stanford University School of Medicine, and served on the Children's Hospital Operation Committee. And she's published extensively in areas of cell biology, immunology, and clinical organ transplantation. Outside of the academia, she ventured into the private sector in 2006, where she was at Stem Cells, Inc., which is one of the earlier stem cell organizations in the private sector that was focusing on FDA-regulated clinical trials of stem cell treatment for children with fatal neurodegenerative disorders. Dr. Millan then joined CIRM in 2012, where she led the formation of the Alpha Stem Cell Clinic Network, which is a clinic of California medical centers that specialize in rigorous high-quality clinical trials at top-tier medical centers for patients that are interested in participating in these trials. And the clinical network successfully currently supports 45 clinical trials and was recently expanded to include 5 programs composed of 7 different medical centers and their affiliated hospitals. All that being said, Dr. Millan, thank you for taking the time to come on the show today.
Thank you so much, Ira. What a great introduction and reminding me of my history.
You've done quite a lot. With that being said, we'd love to hear just a little bit more about you. We typically give the guests on the show the floor for a little bit, just talk about themselves, your background, where you grew up, how you developed this voracious interest in science and medicine, transplant surgery, and ultimately what pulled you into both the stem cell industry, but also this fascinating California Institute of Regenerative Medicine initiative.
Thank you, Ira. You did a spectacular job kind of chronicling my journey, but I wanted to give just kind of a little bit of insight into what led me from one place to the next.
Great.
So I actually come from a long line of doctors. Okay. Back in the Philippines, my grandfather was a village doctor, in a remote area. I had uncles, aunts. It was one of the revered professions, especially in the Philippines, where healthcare, as well as other places where healthcare really is such an important component of maintaining, especially in remote areas where sometimes you don't have as much access to healthcare. So I grew up in that environment, but I actually, in terms of going into medicine, initially fought the notion because it seemed so, in a way, predictable and cliche. So as a teenager, you often don't do what you think you're supposed to be doing. But one of the things when I was growing up, I had a huge interest in science fiction. I loved reading science fiction.
Me too.
My favorite shows were science fiction. $6 Million Men every weekend, watched it. And I was fascinated by the prospect of being able to solve major problems and debilitating, devastating problems with technology and with science.
Mm-hmm.
So that was something that I realized I was interested in. As an undergrad at Duke, I had the great pleasure of taking courses in an early field in immunology. And so things were just starting to unfold then in terms of solving the mystery of how our bodies protect ourselves, typically from tumors and from infections, right?
Mm-hmm.
And around that time when I was in college, the AIDS epidemic had just hit. And so more and more, we were learning more and more about the immune system. In addition, there was progress being made in this very new field called transplantation.
Mm-hmm.
So then I started becoming really intrigued by the science of it all. And one of my mentors at Duke was an immunologist who was characterizing the proteins on our bodies that help our immune system recognize self from non-self, called the HLA. Bernard Amos, one of the early, and what's really great, and maybe it's not a coincidence, he was a surgeon, but he was really best known for his research in immunology. So in those days, as you know, this is in the 80s, it was really quite novel that clinical physicians, specifically surgeons, would be involved in such deep science. So, but I grew up with it, so I knew no differently. Mm-hmm. And then when I went, so after that, I was deciding whether I would in fact go to medical school or continue graduate studies because I loved science, and decided on medicine because of a simple thing. One of my advisors said, "Maria, do you like to interact with people?" I said, "Yes, I love to interact with people." And that's not necessarily fair because I know a lot of scientists who are just the most interactive, and but it was just one of those kind of defining moments when I said, you know what, I'd love to do that because I do think that I could still pursue research as a clinician because of the role model that I had at Duke, role models. And so I did go into medicine. I was very, very attracted to surgery because it was so definitive. Mm-hmm. So I told you I came from a long line of physicians, but I also came from a family where relatives were taken very early by different diseases despite medical therapy. Sure. An uncle in his 20s to seizure, uncontrolled seizure. My uncle in his 40s, he was a cardiologist himself, but died of heart failure. Mm-hmm. So just those examples led me to think we need to do better. And so all throughout my training and everything else, research and finding, asking questions and figuring out where we can answer these questions was always part of the way I trained. And so going to Boston, I was in a perfect setting because Glenn Steele at that time was my chair of surgery, and he is a big thinker. He was a physician, surgeon, scientist who had a very active lab in cancer research. Mm-hmm. But then he eventually ended up going to become a dean of University of Chicago Biological Sciences. And then after that, he ended up at Geisinger Health. We turned the whole health system around through data, through just the use of data, figuring out the best ways, data-driven way to deliver care. So those are my kind of role models and mentors. Mm-hmm. And if just looking back, it's not really. It actually is not that unusual that I would take the path that I did, because just like they did, I followed where we needed to continue to solve problems in medicine. So as a transplant surgeon, fast forwarding through surgical residency, fellowship, and then faculty at Stanford, it was the most gratifying career I could ever imagine, where we were able to directly solve problems, with fatal conditions in babies and adults, where we could turn it around by transplanting the dysfunctional or the organ that's no longer working. Mm-hmm. And it seems so direct, but I always tell everybody it was the earliest form of cell therapy. because
Sure.
all the cells were transplanted. So it was really a proof of concept of how you could turn medicine around and all of the associated morbidities that came with that disease. So going to CIRM after my initial foray into the private sector, the reason I went into the private sector is I was fascinated by how science was turned into the types of technologies and medicines and techniques we were able to apply into the clinics.
Mm-hmm.
And so I was really fortunate to be part of an era where we had some amazing advancements in transplantation and being able to have long-term excellent survival. But we weren't there yet. We didn't have enough organs for everybody. Not everything could be treated by transplant.
Sure.
So the combination of the field of stem cell and genomics starting to really come into being in the late 90s, early 2000s with the Human Genome Project, with the first human genome being sequenced in 2003 and the human embryonic stem cell being isolated and described in 1998.
Mm-hmm.
It was really a convergence of advancements and discoveries that captured the imagination of many, including mine, in terms of could this be the way that we tackle currently untreatable diseases. So 40% of our population, about 130 million people in the US, have chronic, untreatable, incurable disease.
Mm-hmm.
And about 6,000 to 7,000 genetic diseases exist where there is no treatment, and they're pretty rare, right?
Sure.
So just how do you tackle this for few patients? So the idea of the technologies and the elucidation of kind of like what causes the disease on a genomic level and genetic level,
Mm-hmm.
and then now the advancements in the ability to edit and manipulate the genome so
Mm-hmm.
that you can correct the underlying issue. And I'm going to give a quick example of that.
Sure.
Is astounding, right? Because what happens is that it becomes a technology platform-based approach. So now those rare diseases now become an aggregate, a whole grouping that one could think about tackling. So I'm going to stop right there for a second, unless you want me to keep going. Ira, do you have any?
No, it's I want to continue listening. This is great. I was going to ask about this, but keep going because what you were, the concepts of what you are, you envision, and then which areas that you are, what we'll call it, translationally most excited about per your career and what you see as the possibilities, I think is extremely important. So continue. Take your time.
Okay. Because I can speak really quickly and I can say a lot.
Take your time. Take your time. Take your
time. So, for example, sickle cell disease.
Mm-hmm.
All right. So we've known about sickle cell disease and the molecular basis since 1940.
Mm-hmm.
Right? And then since then, more and more specifically that it's caused by a point mutation that leads to a single base pair error that leads to a defect where normal red blood cells, which are disc-like and flexible, that can go through your vessels and deliver oxygen to all the tissues in the body. What happens in sickle cell disease is an inherited disease, typically afflicting African American, but also Latinos and other populations. 100,000 in the U.S. and millions in sub-Saharan Africa as a hotspot. Is that under conditions of stress, low oxygen, these erythrocytes or these red blood cells in these patients become sickled and rigid. Mm-hmm. So they block off the blood vessels, causing extreme pain crises, but also causing little infarcts in different organs in the body, including the brain. And these attacks come on at any time. They're unpredictable. They're debilitating. They lead to multiple hospitalizations, and they lead to high healthcare costs. And the tragic thing about it is not all patients get the care that they need, because it's, first of all, it's rare enough, so not all caregivers would know what to do if they showed up in the ER. The second thing is pain is a major presenting feature. So especially this day of the opioid crisis,
Mm-hmm.
pain often with opioids, and in this case, it's warranted, right? However, now, especially where we're trying to figure out how best to address the issue of there are these patients, in addition, are kind of caught in the crossfires of that. Their lifestyle is completely different than other kids. Usually this afflicts younger adults and children, and their lifespan is shortened.
Wow.
So lifespan today is about 80 years plus, and the average lifespan still is about 40 to early 50s in patients with sickle cell disease. So, and it's right. So now, with that background, the idea that something we've known about, a single typo, a single point mutation, is the reason for this. And now with all different gene editing tools using inactivated viral vectors, we can use viral vectors to deliver the gene of interest, or what's called CRISPR-Cas9, which you probably, and many of the listeners have probably heard about, which is a naturally occurring enzyme where in bacteria use it to protect themselves against viruses. So it recognizes specific areas, and it cuts out specific sequences from the DNA. So using that technology, and there's now next generation even versions of this, one can specifically target an area of the genome and take out the incorrect code and allow correction to the correct code. And in the laboratory, there's proof that there's proof of concept for this, that this could actually reverse the defect. And that approach is now in clinical trials.
Excellent.
And CIRM, and I didn't tell you about CIRM, but I will, California Institute for Regenerative Medicine, where I currently am, is an amazing agency. You described how it was formed. And but I would just like to say that one of the things, and I'll describe a little bit more, the multifaceted ways that CIRM has impacted the growth of this field. But the National Heart, Lung, Blood Institute, launched a Cure Sickle Cell campaign, where the idea is to challenge, we're going to cure sickle cell disease. And because of CIRM, the type of programs we have to accelerate the research, the processes, and the type of in our ecosystem of scientists, they partnered with CIRM, and we are jointly funding programs to increase the potential approaches. We don't know who the winner is, but there are some very promising ways to target sickle cell, for instance.
Excellent. Along those lines, and definitely, I'd like you to take, because I know a little bit about it, but I think the audience would really like to hear about it, because CIRM, obviously you've had you've spent time in academia, you've spent time in the private sector. CIRM's a little different because you had to go out and the propositions and government involvement and taxpayers and so forth and so on. Walk through a little bit about sort of the history on how this all started, and sort of, I know you took the CEO position a few years ago, but what are your responsibilities as CEO? Obviously, you have to do more than oversee the lab. You have to give out grants. You have to, oversee research at various laboratories throughout the state, which is a huge state, and you got a lot of great research going on there. Talk a little about what you do on a daily basis, what your day is like, and if you could give some background of sort of how CIRM started, and sort of some of the challenges of getting it off the ground, and sort of the voters, all that stuff. I think that's really important part of the story.
So CIRM was created in 2004 through a citizen-led bond measure. And I had described earlier that the human embryonic stem cell was first isolated and described in 1998. So that was just a remarkable discovery, because it was shown that all the cells and tissues in our body arise from a single ancestor, the human embryonic stem cell, and that these cells can be cultured in a dish, have unlimited ability to self-renew, and then if you give it the right factors, the right growth factors and conditions, it can be differentiated into any cell in the body. So imagine that. So a couple of things. That opens up all sorts of possibilities in terms of how that could be used for studying disease and studying what could potentially work, called disease in a dish. So whereas it's not easy to get a brain biopsy per se from somebody afflicted with a neurodegeneration disorder, one could envision, and it's being done, you could take these embryonic stem cells, differentiate them into various sorts of nerve cells, and then test that, right? Another application, which is even more directly related to where I started off, which is actually the use of these cells as potential replacements or treatment for tissues and organs that have gone awry and are no longer functional or are missing necessary function. So because of this promise, the California citizens wanted to make sure there was a reliable source of research to pursue the science to address these unmet medical needs. And in this case, embryonic stem cells seemed like a really important new finding to pursue. Around that same time, there were restrictions on federal funding for embryonic stem cell research. And by necessity, there were ethical debates also occurring at the same time. Okay. So by Californians passing this legislation, it's called Proposition 71, it led to a reliable source of protected money to specifically pursue science. That is a historic and one-of-a-kind state-led initiative.
Absolutely.
And so because of that, CIRM has been able to fund over 1,000 different programs. I can describe those to you. And now, in the first 10 years or so of CIRM, some of the questions were, could we even take some of this? Would the technology be something that could then be tested in clinical trials and developed into therapies, right? So in the first 10 years, there were, 10, maybe 15 initial clinical trials that were just getting started in the early phases. And today we have 60, we funded a total of 60 first-in-human, mainly clinical trials. And I'm going to get into that a little bit more, because you had asked what are some of the things we do on a day-to-day basis to continue to accelerate this field. So for that to all occur, the pipeline starts with discovery and basic science, really solid science. And then, so we fund discovery research, and we've invested about $900 million into basic laboratory bench research to really hone down on the biology, the genomics, the characteristics of potential approaches to disease. And then it goes to this phase called translational, which means you take the science and figure out how you can predictably make this candidate product, and then put it through the type of testing one would need to be able to go to the FDA and say, now I want to test this in clinical trials. Science is something that typically is not funded by the NIH. They do some, but typically that's a type of thing that happens in a company once they've already decided they're going to invest in this. So think about this situation. It's a very new field. Most companies and investors, they're not going to take a risk on it, right? Because they're going to put their money into traditional drugs, using all of the traditional ways that they do the drug development, or antibodies, where they know how to do this. So the role of CIRM is what we call to de-risk the programs. by investing early when we know there's solid science to do the type of things that maybe couldn't be funded by traditional funding, either through industry or NIH, and then to take it to the point where then it can be tested in the clinics. And then the type of clinical trials that are run out of these type of programs are unique in themselves. So you had mentioned that one of my first projects that I was involved in was forming this Alpha Clinics network.
Right.
And one of the major reasons we funded formation of this network, academic laboratories and hospitals do clinical trials all the time. However, there's some unique aspects of doing these types of clinical trials, which really requires a certain level of expertise.
Sure.
So we wanted to be able to develop that expertise so that we can continue to export that and bring others along and build upon this network. And that has been ragingly successful. In fact, our Alpha Clinics have told us that since they formed the network, they were able to attract not only the CIRM-funded trials, but when industry was starting to develop the trials, such as the CAR T technology, which you may have heard about, which are the engineered T cells, those were the types of centers that they needed, because again, those were cell therapies, and they have very unique characteristics and needs versus traditional drug or even antibody type trials. To make this all happen, we have a team of internal scientists and professionals in various fields. CIRM is very unique because it embeds within it, first of all, the patient is at the center. Our mission is for the patients, patients with unmet medical need. Patient advocates were the driving force behind formation of the agency. Patient advocates and representatives are on our board. They're on our advisory panels. They join us in our workshops. They organize ways that we can discuss many important issues with the public and scientists all in the same room. We have scientists in-house who help to manage these awards. So they're almost part of the team. You know how in venture capital firms, they have, kind of executives and residents. That's what our scientists do. They are part of the teams that we fund. But we don't just do that ad hoc. We've actually formalized that structure and have formed advisory panels called clinical advisory panels or translational advisory panels, where we bring in experts from outside of CIRM. We have the CIRM team, and then the grantees, the ones we're funding to do the translational or clinical research. So we can make sure that we problem solve along the way, because these are not easy projects. Right. Folks stay on track. And the cool thing about that is we've seen that type of system has allowed us to increase the probability of the tasks being done in the more timely fashion. And after all, our mission is to accelerate where we can without compromising safety or rigor, to accelerate the development of this science so that it can get to clinical testing and then to patients. So the purpose of the panels and the purpose of our scientists being so involved in this is to do just that. Another component of what CIRM does, we by nature are a funding agency. Right. But the thing about how we fund and how we run things is that it's built to accelerate. So everything we do in service of the mission, and then my predecessor, Randy Mills, very business-minded and really just brought focus and a structure to the agency, really led us to drive an efficient machinery where we would have a predictable funding so that people came in when it was the right time. They didn't feel like they had to force fit their projects into, a random, unpredictable access to capital. And also, we accelerated the time from the applications coming in, and if they went through our peer review and then our board approval, they could get their money within less than 150 days, which is one of the fastest out there in any type of setting. So even down to the details of how we do business, we're accelerating. So accelerating through the quality of the science, because we put all projects through what's called peer review.
Sure.
So we have a panel of experts from outside of California, just the leaders in their field, and from various areas, scientific areas, clinical expertise, manufacturing, regulatory, business, and taking a look at these projects from all those vantage points.
Mm-hmm.
And so, and internally, we have teams that look at it from the business perspective. We have a business development team that tries to optimize the visibility when projects are going along to the business community so that they have the chance to come in and support these programs and form strong commercialization partners to take it down the road, because CIRM doesn't have unlimited funds.
Sure.
And so the idea is to de-risk, help them to a point so that they can gain these partnerships, because then the rest of this getting to commercialization is very expensive. And we've actually increased, early on when we started the current strategic plan is a 5-year strategic plan.
Mm-hmm.
And the goals of this plan was to accelerate, build a pipeline, improve and refine the regulatory paradigm. And I'll give a little bit of detail on that in a bit. Increase the portfolio of strong clinical programs to address unmet medical needs. And the fifth thing is increase the probability of these programs being partnered with industry and investors.
Mm-hmm.
Because early on, there was very little industry pull. Now we're seeing so much more. So when we launched this, we were seeing, in terms of publicly available information, maybe $50 million in industry investment in partnership into our program. The next year was like 120. It kept going up. And then the past years, it was over $1 billion each year. And so about $3.4 billion that we can capture of now industry investments have gone into the programs to continue to bring this along. And some of the companies that came from programs that started in academia then spun out, and then we continued to partner with them to get through the early clinical stages into the public market, had successful IPOs and have been able to get capital from that. So it's, as you can see, it's the idea is it's taking all of the various aspects of what it takes to take the science and turn it into something potentially therapeutic, taking into account the business side, making sure that the patient, that we keep patients in the center, because it's so unbelievable how valuable even early input from patients are, because everybody gets really excited about the science. Everybody gets excited about data, trying to get data. But then when you think about it, it needs to go to patients in the real world. What are going to be the important things to them? And that's something that's going to be important as we go forward, because it's not, in terms of the value of these therapies versus the initial cost, because these therapies are going to be expensive initially because of the way they need to be manufactured. We need to have all the information to show what is the impact, the true impact that it has on the health and the life of patients who've been treated with these transformative therapies.
Absolutely.
Go ahead.
It's really remarkable the scope of what you're involved in, and I think there's as you were saying with the de-risking, I think it fits very nicely with this sort of what is referred to this widening valley of death that we see throughout biotech, about how so many folks are so fearful of the early stage stuff that it gets forgotten, and I think that you've played an extremely important role in filling that gap. I want to go, you mentioned something, I want to sort of go to now the other end of the spectrum in regard to that word regulation, because obviously these are, well, not all of them, but many of them are living cell products requiring, unique regulatory guidance and so forth from FDA and other regulatory agencies. Yet, sort of parallel to all this, as the research has been going on and clinical trials have been happening and so forth, we see sort of a very unique state of affairs happening elsewhere, in the sense of Japan with their conditional approval regs, and China has developed some of these special purpose medical zones lately. Obviously, the dynamic of medical tourism has been flourishing. How does a lot of that fit into sort of where CIRM, the ways that CIRM is thinking in terms of, obviously the U.S. is an important space, and this is going to be your main thrust in terms of regulatory approvals, but how are some of these other regulatory domains around the world sort of affecting what's going on at home here in terms of California? Are any of those dynamics directly impacting sort of the way you're doing things on the regulatory side, or it takes a little bit in that direction?
Absolutely. I mean, we're all, this ecosystem is all interconnected.
Mm-hmm.
And so I'll just back up by saying one of the major advancements that we've seen in the regulatory component of this is the passage of the 21st Century Cures Act in December 2016. That created some resources, but a motivation and actually a declaration that the FDA would commit resources toward evaluating how to bring these regenerative medicine technologies forward.
Mm-hmm.
And so they created what's called the Regenerative Medicine Advanced Therapy designation,
mm-hmm.
which is kind of a combination, has features of expedited programs that the FDA has had before, but it's specifically for cell and gene therapy programs.
Okay.
And a couple of things were happening at that time. Scott Gottlieb was the FDA commissioner when this was all being launched. And one of the things that they did, or a lot of the things that they did, is the FDA brought more clarity to a lot of fuzziness that was out there. And
mm-hmm.
it's continuing to work on that in terms of guidance documents and approaches toward doing the type of research with these complex type of technologies, as you had mentioned. There are mm-hmm. very aspects to these living medicines. And we have seen a really remarkable, not that the whole thing's done, but a learning environment that creation of the RMAT designation and all of the things that FDA have done to increase the transfer of information and trying to get in on the same page in terms of what the FDA needs to see to be convinced that a product is not only safe, but is something that has an effect that they could say they could give an approval to go ahead and get that marketed, right?
Mm-hmm.
One of the kind of the unique features of the types of programs we're talking about is, as I mentioned earlier, they often afflict small populations.
Mm-hmm.
And the initial indications, especially, are for small populations, often, in the extreme ends of their diseases.
Mm.
But the remarkable thing is that because these cell and gene therapy approaches are the mechanism of action typically is pretty well defined. You're targeting a specific thing to get
mm-hmm.
an output. What's called the effect size is something that's usually larger than one would see in some of the traditional drug development, right?
Sure.
So the effect that you're seeing is much more definitive. Well, what that allows is that with a smaller number of patients, then you can get a bigger effect. And so, the statistics of that are in favor of being able to make more conclusions with smaller numbers.
Mm.
And so, but that also means that all the data is extremely important, and it also means that this opportunity to have frequent interactions with the FDA as you're going along and getting this information is crucial. One of the things that we're seeing, to just let you know, I mentioned that we had 60 clinical trials,
mm-hmm.
that we actually have a handful that are now in what's called pivotal, the
mm-hmm.
pivotal stage.
Sure.
In classic drug development, usually the pivotal stage are phase 3 large trials, controlled studies with control arm. We're seeing more and more diversity in the type of trials that are reaching pivotal stage.
Excellent.
For instance, We have a program that's one of the furthest along in an immune deficiency, a primary immune deficiency called ADA-SCID, bubble baby disease.
Sure.
That's SCID stands for severe combined immunodeficiency. And it's caused in this case by a missing enzyme, adenosine deaminase, which does not allow these kids who inherited this disease to form immune cells.
Mm-hmm.
So Don Cohn from UCLA is the primary investigator who developed this, uses lentiviral gene delivery to deliver the missing ADA into the blood stem cells that are recovered from these kids from the peripheral blood. Mm-hmm. These blood stem cells that are corrected now for the enzymes are returned to the babies, and they home to the bone marrow, where they do what blood stem cells normally do, which is give rise to all the blood cells of the body, mm-hmm. including immune cells. Okay, so I mentioned effect size. So one of the patients that we've shown in our annual report is one of over 40 or 50 who have been treated and over 3 to 4 years ago, back. And Evie, who's been on our annual report, is now, I think, 6 years out. And she, from the time she was treated and they started to see effect to today, still makes normal immune cells that can be detectable in the blood. So the idea that she was born without an immune system, immune cells, and you detect it by doing a blood test,
sure.
is one of those things that doesn't, it's very concrete. And also you can measure the enzyme that's replaced. So those types of approaches lend themselves to those types of outcomes, in addition to other things such as, the standard lifespan, what's their survival, and all of the other complications, other clinical parameters. But then you also have these laboratory concrete data points. Mm-hmm. So we actually also have other programs where they're in early phase 1, 2, and because of conversations with the FDA, have been able to get really great advice in terms of how they can even modify their trial, and they're allowing them to do this. So this is called adaptive trial design.
Sure.
In the course of doing the trial, so that they can get the best information, and then that could actually lead to them getting earlier approval if they get that information. So it really has turned the regulatory paradigm around. That I mentioned that idea was refining the regulatory paradigm. And CIRM, in partnership with the FDA, in partnership with their investigators, have been able to obtain a very good, initially the first, among the first RMAT designations, CIRM had about 30 to 40% of them. Mm-hmm. And even today, CIRM has 6 of the RMAT designations of approximately 40 something, I think, that are out there today. Mm-hmm. So the type of programs we fund are very well suited for this regulatory paradigm. And you had mentioned some conditional approvals and other models in Japan, for instance, mm-hmm. where products might get an approval with very little data, then they then submit data later to support that this is something that they will continue to support being out there in the marketplace. We don't have that, but what's happening with the type of programs that I mentioned is that the FDA is willing to look at approval on small numbers initially, but they will require post-approval data to come back to continue to support this. So, because we want to make sure that when it's released out there in the, to the more broad population of patients afflicted, that it validates the same, safety and efficacy that we saw in the clinical trial subject. Mm-hmm. So I hope that answers your question. We have a different form of accelerating the development, and the really exciting phase that we're in is that we still have a challenge of how we can align how we're developing the therapies, getting it through the regulatory paradigm, but then making sure we get coverage for this, once it's approved. Right. And so the data sciences is going to be really important in terms of integrating, not just designing the trials and the follow-up in a way that supports approval, but also supports access to patients by justifying the cost of it and the coverage of it for these given patients.
Right, absolutely, big part of the puzzle. And I appreciate you, that overview of what's going on with Unmet, and I think that's extremely exciting. Especially, so many people complain about FDA, but they really do. When it comes to some of these unique areas, they really do step up and try to work with companies and organizations to get stuff done. And I think that's an extremely positive sign and a positive thing that you bring it to, as you said, ecosystem.
We're in a very collegial and collaborative phase with the FDA.
It's important.
In this field. One of the things you had mentioned, and I didn't want to let it pass, is the whole problem we have with stem cell, what's called stem cell tourism or direct to consumer.
A lot of that going on.
It's a big problem. It's a distraction because, as you know, we've already seen some horrible complications of patients who've gone to these so-called clinics where treatments are being offered for a variety of different conditions, from baldness to Parkinson's and Alzheimer's, with quote unquote stem cells, which are non-characterized cells. They just kind of hijack the name, right? Taking advantage of the fact that there's exciting progress in the science, hijack the name, not been through any rigorous research. I mentioned how we put programs through rigorous research, rigorous evaluation, then we have data-driven, ways to determine whether it goes forward. These have not been through that, and they charge exorbitant prices for people to get these uncharacterized treatments, and then don't have follow-up. It's really horrendous, but it's a huge industry, as we're becoming aware. The FDA is making some initial progress in enforcing and going after some of these bad actors, but there's still much more that needs to be done. I think that we need to what is CIRM doing? What we have, we play a support role, and we give input, and we provide education, but also are involved. Our team gave input into the Hernandez bill in California, which was had gone through, which requires that clinics who are claiming to deliver these therapies, if they don't have FDA approval, they're required to post that and to give that information to patients. It's not enough, but it's a starting point.
Yeah, it's a starting point.
We are really looking for ways that we can continue to show the contrast between legitimate work to these to the rogue work. But the good thing is, the more examples and instances we have of legitimate treatments that go through the standard ways one delivers, treatments and get insurance coverage and everything else, the more I think the public, along with our help, we're really need to do better in terms of education and getting the information out there and helping to guide them through the whole thing, so-called navigation, which is definitely on our radar. But the more there are instances of legitimate treatments that went through the right ways, not necessarily in that particular indication, for instance, but that it is possible. I think the more we'll be able to help the public and practitioners determine the difference between legitimate and non-legitimate. But it's not by any stretch solved. We still have some, on multiple fronts, work to do. Sure,
but it's a beginning. You've mentioned a few rare diseases in terms of the sickle cell and the SCID. Obviously, you, as you said, your budget is not unlimited, but I'm very interested because if you just look at that rare disease list, there's I don't know what the 7,000 or something different conditions on that, representing something like 400 million people around the world. What do you? I mean, if I got this question out there, if the sky opened up and there was unlimited money sitting around, What are you going to be doing for the next 20 years? Do you want to focus mainly on the rare diseases and conquer them? Are you more interested in the more traditional, the Alzheimer's and the congestive heart failure? I mean, what really excites you, and what's the stuff that keeps you up thinking the next 10, 20 years out, where you would like to put more of the resources? All the above.
All of the above. And Ira, I noticed that one of your previous guests was Victor Zhao from National Academy of Medicine, and he talks about the idea of not just extending the fact that we are seeing a remarkable increase in our longevity.
Right.
It's not just longevity; it's healthy longevity.
Yep.
And so the longer we live, the more we are going to be afflicted by different types of these are cancers of aging, cancer, Alzheimer's, all sorts of neurodegeneration, cardiac disease, right? These are metabolic diseases. So they all are important. And actually, I focused on rare disease maybe in this conversation because they are, instances of where we have gene therapy approaches, and they're kind of measurable.
Right.
There are the more complex diseases we still can target with cell and gene therapy. They're just more complex, and so they're just we need to have fit for purpose approaches. However, we have made such progress that we can also apply the general knowledge to it.
Absolutely.
So we also fund programs in type 1 diabetes, and we're seeing some I don't know if you know about this program, but it's embryonic stem cell derived pancreatic progenitor cells that when planted, the idea is that the cells would give rise to insulin producing cells, which are missing in type 1 diabetes, the beta cells. So ViaCyte is a company down in San Diego that has been funded by us, and they reported recently on 8 patients who have been implanted with this combination of device and cells that have produced insulin whereas they didn't before. It's early days, but they're pushing along that. And what's really exciting about that is once we get proof of concept, we can continue to layer on more of the advancements to tackle the challenges of making this more durable. So what ViaCyte's done is also partnered with Crispr Therapeutics, which is a gene therapy company, to remove the antigens. I mentioned my early interest in research in immunology. The antigens involved in rejection, being rejected by the body, to increase the chances of these cells surviving once they kind of establish themselves and become insulin producing cells, so that they're accepted as self, or so that bodies can be effectively tolerized to it. They're also working with device companies such as W.L. Gore to get even better materials, the durability. And so it's just an example, large indication, and a transformative approach.
Absolutely.
But how do you continue to bring in the technology to even make it, we're already, while these technologies are being developed, next generation approaches are already being introduced along the way. Which is unbelievable. We also funded programs in neurodegeneration, but as you say, we still have much more to do in that space. But one of the advancements one of the things we're thinking in terms of the future is, how do you gather all the information? There's just a wealth of information, genomics data, clinical data, that have come out of all these attempts to tackle Alzheimer's and various Parkinson's, Huntington's, but there is a convergence of the advancements in science with advancements in technology and computational power. And so how do you do that in a way that you can all continue to more deliberately identify potential targets that you can then go after with all this amassed data? So one of the things that keeps me up at night is, how do we get all the major stakeholders who've just gotten this wealth of data all together, and so that that can be made accessible to our scientists and our researchers, so that we can continue to be a learning environment where we keep getting better and better in terms of how we're approaching problems. And my team and I were just at in the Santa Cruz campus, UC Santa Cruz, yesterday, as a matter of fact, with David Hausler, who's one of the scientists who led, who was on the leading team for the Human Genome Project.
Mm-hmm.
And their team has just, kind of updated us on the remarkable progress they've been making. We've funded programs at UCSC as well as others around the state and our genomics program. But they are very much in agreement that there is a way that all of these various knowledge sets that we can, look to how we, in the future, we can bring this all together to tackle these vexing problems.
Absolutely. You brought something else. You stimulated one other question, and you've given so much of your time, so I don't want to take so much of it. But this one, you awoke something, and I just want to throw it at you to get your feedback, because you mentioned Victor, and when he was on the show, we were talking about the longevity challenge at the National Academy of Medicine. It's a $30 million program. I also did a show with a similar counterpart, George McGuinness in the UK. They have a $125 million program focused on this topic of sort of healthy longevity and sort of ways we can impact things a little upstream from the pathology, sort of some of the maybe the higher level drivers, let's say, of different aging processes. In many ways, what's going on in sort of the longevity biotech world, I guess, is similar to what was happening in stem cells maybe 20 years ago, where you had some insights, but there's still going to be a valley of death. It's going to take a while before you get a lot of people really wanting to invest a lot. Now, you've had that experience, $3 billion, you've been doing these amazing things with it. Are there any learnings from this model, from, I know you're probably raising more money in the future, but sort of this public-private model that may apply to other moonshot ideas like aging, in the case of some of these other projects where, the venture capitalists don't, it's not prime time for them yet. Any tips for other people that will be listening to the show that they want to do what Maria did at CIRM, but we want to do it for aging, or we want to do it for, going to Mars, or whatever it may be. Any tips along those lines?
So the thing is this, that's funny that you say that, because I am a tiny little piece of this whole machinery.
But you run it.
And that's the secret, is that it needs to be multi-stakeholder team approach. And it's also, it's if I just look even at my time within CIRM in terms of how we iterated and passed the baton and then learned from what worked and what didn't work, and then created then more efficient systems to address that. One of the things is multi-stakeholder. It can't just be one section of our society working on this. We all need to be together on the same page, understanding what it is we're trying to drive, right? We need to think about not only the scientific aspects, but the social aspects of it. And it's not just to be fuzzy, it's for real. Because when I said data, think about every aspect of this has the public involved, because the public owns the data. We have data issues, and meanwhile, data is going to be the key, right? So that's going to be, those are huge things to tackle. Policy, advocacy, scientists, engineers.
Yep.
But the key thing is collaboration. The NHLBI CIRM partnership is a great demonstration project of how the NIH, which is the largest funding organization, which is, just has such a lot of different systems even within the NIH. They said, we want to tackle this, and we see that we can do this more efficiently in partnership. And CIRM said, yes, let's make it happen. It can be as easy as that. And to take complex systems when you realize, and so the key thing is, we all are around a mission. In that case, is to cure sickle cell. CIRM's broader mission is to accelerate treatments to patients with unmet medical need. One of the instances is sickle cell. So everything, we can always keep going back to that mission. And the idea that I think, so I came from academia, as I had mentioned, and one of the wonderful things about academia is you have all these brilliant minds and all these ideas come from it. One of the major hurdles in academia is the siloed approach.
Yeah.
It's starting to improve, but it was just one of those things I observed. I felt like, wow, we could actually solve this issue if we truly, and because we say, oh yeah, let's collaborate. Maybe you'll get this material. You try to get the materials transfer, and then no, because this person wants to publish a paper. It's crazy stuff. And the thing is, it's the whole culture of publications and open source data and all that. We are starting to see that whole movement is occurring, and I'm just so pleased, because that is one of, to watch things not go forward because some of our weaknesses as human beings get in the way. We let it get in the way. That's frustrating, because we, especially if your whole reason for being is to solve problems and make things better for the world. And I think all of us are doing that in our own way. I think that you want to overcome that. Most people do. It's just providing the right guideposts for that. So at CIRM, we do have motivation, which is funding, which is money, and it was up to us to say, okay, here are the conditions of how we will make this accelerating. So make it less painful to get the money so you can start the work, but there are some requirements that you're actually going along that path. And what's great about it is people do want to go along that path. So I guess structure, staying mission driven, and collaboration, not just for the sake of collaboration, but because you truly are trying to tackle a given problem.
One last question while I have you. You mentioned early on a few of the sort of the influencers that have guided you throughout your career, and you've no doubt met dozens of very fascinating people in academia, in industry, in government. Any specific people at this point that you want to shout out to, that specifically that if it wasn't for them, I'd be talking to Maria Millan, lawyer, right now, or Maria Millan, real estate tycoon, or anything else besides this?
I mean, I have to say, it was what I get asked that question, and honestly, it's hard to really narrow it down to just a few, because I've really been fortunate along the way that I can even remember why I went into surgery, because Ben Rush, who was like one of the old one of the kind of the icons, early surgeons, just looked over at me and he just said, "You should consider surgery." And I never even considered it until that day. I said, okay, I'll start paying attention in the OR. And I realized, you know what, it's compatible with my personality. So it's people who kind of recognize, oh, okay, I see something in you that, that seems like it'd be good for this particular thing. Or when I did my research with Fritz Bach, who is also a very well-known immunologist and transplant immunologist, he was a great mentor for me, because he just everything's possible. If there's a question, you don't, you get away from thinking, well, that's not what it says in the textbook. So just kind of opening your mind. Judah Folkman at Boston Children's Hospital. During my surgical residency, we used to make rounds, and we called them Folkman rounds. Just for everybody who's listening, Judah Folkman was the scientist. He was also a surgeon, pediatric surgeon, Boston Children's Hospital, Harvard, who identified VEGF as being the factor that led to tumors proliferating through observation.
Right.
The clinical observation was that when you remove the primary tumor, all of a sudden these micro tumors that were out there in the rest of the body just proliferated. And he said, there's got to be something in that. So he started isolating factors from urine. And then one of my own really good friends during residency, Kathy Chen, was in his lab during that time. We all did lab time when we were in training, was injecting various components that were derived from these models into the cornea to determine the vascularization, and that
Mm-hmm.
so these seminal discoveries that came from from a very like tangible clinical observation, then led to this, and then it led to, of course, Lucentis and other kind of
Right.
approaches to VEGF. So Glenn Steele, I mentioned, big thinker, all the way from the bench, OR, then transforming a whole healthcare system, and he's still a leader in thinking through this. I've just been really fortunate, to just interact with all these folks. But you know what I wanted to end it with is every day with all these different investigators that I have the great honor of working with. They are all role models. I mean, every single one. I can't even cover all the progress in a single day of all of the amazing work that's being done, and their teams, the unsung heroes, the behind the scenes people who are figuring out the postdocs and the scientists at the bench, along with these clinical researchers who are bringing it to the patients. They're remarkable. I just sit there and and just talking to them in terms of their own journeys and then what they're tackling now. The remarkable thing is people don't sit still. They don't just rest on the laurels that, oh, I became famous because I had this discovery. No, they're, if you see most of these people today, they're still tackling the next problem. So I think that I've just had the great fortune of being surrounded by such input, and then the patients.
Sure.
The patients are truly motivating, and patient advocates and the folks in the community who are tackling these issues from a variety of different angles. The social determinant aspects of disease and all these things that are so important when we're thinking about, we're not doing this in isolation.
Right.
And so anyway, it was really wonderful to I'm happy.
You're the first person that everyone I ever brought up the patients, and it's a big piece of it.
It's the reason we're here. It's the reason we're doing this. It's the reason I went out. I didn't get to say, but when I used to transplant these little babies, it was the most gratifying thing to be able to do this technically, complex procedure, and then to see them get better, and then to manage them in the ICU, and to manage your immunosuppression. Just so I was able to put in the science, the technical, the critical care aspects all together. It was so gratifying. But then I was also there when I realized, just seeing them go through these extended, hospitalizations, and these families by their bedside, and the moms who often were the donors for these living donor liver transplants were also patients. They had just given birth 6 months before, taking their left lateral segments so that their babies could live, right?
Yep.
So just looking at all that and what they go through and thinking, well, okay, we can do that now. We got to get to the next stage. How can we do better, right? How can we do more? And that, for more people. So that was what motivated me to continue to pursue the next phase.
And I'm glad it did, because you really, as you said in your bio earlier, you've devoted your career to treating and developing these different technologies and products and solutions for these unmet needs. And you speak with such passion about it, and it's just, you've had a fascinating career. I wish you the best keeping it going, and
there's only
the next stage.
One of the things is I always wish I think I read somewhere, it's like, oh, this is so remarkable what we're going to be able to do. I wish I could live long enough to continue.
You never know. You're working on. This is all healthy longevity.
It's passing the baton. We have so many amazing people in this world working on various aspects of this, Ira. Wow. And for you to communicate this to the broader community, that's an important piece of it. You play an important role.
Thank you for that.
So thank you.
Thank you. And once again, for everybody watching on the YouTube channel and listening on the various podcast networks, we've been talking to the amazing and inspiring Dr. Maria Millan, who is the President and Chief Executive Officer of California's $3 billion CIRM initiative, California Institute for Regenerative Medicine. Doctor's devoted her career to to treating both children and adults with these major life-threatening diseases. As we say on the show, moving the human story forward. Maria, thank you for not just coming on the show, but thank you for everything you've been doing the last few decades and continue to do. And it's really been an honor having you and listening to your story. And thank you, and wishing you the best of luck moving it all forward. It's really a great thing.
Thank you so much. It's been a pleasure.